DNA replication. For this lesson, we're going to talk about the DNA replication, what it is. We know DNA replication means to copy the genetic information of a cell. Before cell division happens, the cell would need to replicate, it would need to copy the genetic material from a single cell before the cell divides into two cells. So the objective of this lesson is we want to summarize the processes involved in the DNA replication, and we're going to describe the role of enzymes in the DNA replication. Replication means literally that when something multiplies, when something continues to increase in number. So replication copies the genetic information, and it's only in biology where you say you have a cell division, cell divides by increasing the number. So cell divides by multiplying. So it's only in biology that the cells would divide by, the cells will multiply by dividing. So cells multiply by dividing in biology. So a single strand of DNA serves as a template. It's going to be a template for all the new strands. So you're going to have remember that the rule of the base pairing, whereby you need to have a T with an A and a G with a C. So this is for sure, you need to know that. So the rule of base pairing direct replicating. So the DNA is actually replicated, the DNA is quite copied during the synthesis phase. Remember that we've got phases before the mitotic phase, whereby the cell is being copied. So each body cell gets a complete set of identical DNA. So this is exactly what I'm talking about. So you can see in the S phase, the S stage is the stage whereby the synthesis, the copying process happens before the division of the cell will take place. So protein is known to carry out the process of replication. The major reason why cells replicate is because they want to increase the number. So the DNA serves as a template. So the DNA will be the foundation or the fundamental on top of which all the DNA will be formed on. The meaning is it's said to be something called semiconservative. That is to say, if you want to have a cell division, you have to get half of the DNA copied, then you have to pair the half with another half. So this is what this is what we mean by having a template. So the enzyme and all the proteins do the actual work of a replication. We have an enzyme which is called a helix. Is the one that actually helps to unzip. It helps to unwind the strand, the double strand of the double helix. So when it unwinds it, then we've got free floating nucleotides, which is formed from hydrogen bonds, which is put within which is poured within the template. So what would happen is this free floating nucleotide bases. These are the free floating nucleotide bases here. So the free floating nucleotide bases are actually thrown to be in between, to be among the process. So once the helicase is unzipping the double strand, then what would happen is the free floating nucleotide would be finding a way to stick with the complementary bases. What do I mean by complementary bases? I'm talking about the TACG pairing. So this is exactly what we mean by complementary bases. So now the free floating nucleotide will stick to the complementary bases, and as they do that, they're forming a new DNA strand. So now a new DNA strand is forming from here, and another new strand of DNA is forming from here. So by the time they finish they complete the process of the pairing, you're gonna have two DNAs from one single DNA. So this is exactly what replication is. So now DNA, there is an enzyme called the DNA polymerase. The DNA polymerase is an enzyme which bonds the nucleotides together to form a double helix. So it bonds the nucleotide, the free floating nucleotides, it bonds them to the complementary bases, which is on the templates of the DNA which we already had. Remember, the DNA is double helix. The double helix is unwinded. So when it's unwinded, then you're going to have a single helix. Remember, we put some free floating nucleotides. When you have a DNA being unwinded, there will be some open or some exposed DNA bases. So the free floating nucleotide will look for the complementary bases and will pair with it. So once they pair with it, you're gonna have the double pairing taking place at the same time. So the polymerase would be the one to help to stick them to the complementary bases. So the polymerase enzyme will form a covalent bond between the nucleotide in a new strand. So this is exactly what we're talking about. So this is the DNA polymerase, which is forming a complex in the location where the complementary or where the pairing up is happening. So when you talk about a complex, you're talking about creating an avenue or creating a room or creating a space for the sticking together of the new nucleotides to the template of the DNA which we already had. So this is exactly what the DNA polymerase does in the replication process. So now after the process, you've got two new molecules of DNA are formed, each with an original strand. So the original strand is the one in blue and the new strand is the one in brown. And this is the reason why the DNA replication is said to be semi-conservative. Semi-conservative because you've got half of the original and you've got half of the new. So this is the reason why it's been said to be conservative but semi-conservative. Replication is usually fast and accurate. DNA replication start at many points in eukaryotic organisms. Remember, we've got two types of organisms, meaning that we've got prokaryotes and we've got the eukaryotes. If you could still remember in the lower grades, we mentioned the differences between the prokaryotes and the eukaryotes, and we said the prokaryotes are organisms that have they don't have a nucleus, neither do they have membrane bound organelles. So the organelles they are naked organelles. They don't have a membrane which is covering them or protecting them. And for the eukaryotes, they have a nucleus and also have a membrane bound organelles, like for example of eukaryotic cells have mitochondria. They also have a chloroplast. Mitochondria has a membrane, chloroplast has a membrane. So all of these are examples of features of the eukaryotic cells. So take a look at these example of eukaryotic chromosomes. The points of replication is not only one. So this is the reason why it happens faster in eukaryotic organisms than in prokaryotes. And in eukaryotic organisms, you can have more than one single replication taking place at the same time. So this is the point now, the replication is happening here and it's spreading out. And now you've got you got two new DNAs whereby you've got one original and one new DNA. So this is exactly what we need to know about the DNA replication. And after the process, the DNA polymerase would have to find and correct the errors. What do we mean finding and correcting the errors? Is gonna run through the process. Is gonna actually check every DNA basis or nucleotide basis if they are accurate. Because if there is a mistake, if there is a frame shift or whatever happens, there will be a wrong reading and it's going to cause a wrong protein being formed, which can lead to mutation. So that's the reason why the DNA polymerase would need to find and correct the errors before the process of transcription is actually started. Now the next one is called a transcription. Transcription is a process which is where you say you transcribe something. The meaning is you're writing something down. That's a linguistic meaning of transcribe. When someone transcribes, mean he writes something down. So transcription would have to convert, it have to change a gene into a single-stranded RNA molecule. In the beginning, I mentioned to you the reason why we have to transcribe, we have to translate. All of these processes we're going through is for me and you to be able to produce a protein, which is needed in the body. And as you know, DNA cannot produce protein directly. So it has to change the DNA to RNA molecule before it can be translated. So that's the reason why transcription is a process of changing the DNA to RNA molecule for the process of translation to happen. Let me cite a good example. You want to buy some stuff in the market, and this guy says, no, you know what? I don't take coins here. I take only papers. And you've got only coins with you. You've got coins of, let's say, for example, you've got $5 in coins. And are you gonna say because you don't have paper, you're gonna have to go back home and you're not gonna buy some stuff? You've got to find somebody who's got paper and give him some coins and take the paper. So this is exactly what happened. You want to buy the stuff, or you need to buy the stuff. You got the money, but the guy is not taking the what is not taking coins in the papers. So you would change the money with you would change the coins into paper and buy what you have to buy. So this is exactly what happens in transcription. The DNA wants to produce a protein and is not capable of doing that until it becomes an RNA. So it changes from a DNA to RNA, and that process is called transcription. So RNA carries DNA instruction. So this is called a central dogma. So the central dogma says the processes of producing protein is a process that cannot be altered. What it doesn't mean, it means that the DNA would replicate itself to form another DNA, which I already explained to you before, a few minutes ago. Now the DNA will now be transcribed to become RNA. All of this DNA DNA replication and RNA transcription, it happens in the nucleus of eukaryotic organisms. But for the prokaryotes, all of it happens in the cytoplasm. But for the translation process, it happens in the cytoplasm in both the eukaryote organisms and the prokaryote organisms. The reason is basically because the ribosomes, the materials which are needed in the production of protein, is found in the cytoplasm than in the nucleus. So the nucleus doesn't have enough material, which is actually needed for the process of translation. So that's the reason why the translation happens in the cytoplasm, where the cell would have enough materials for that process to happen. But the process of translation, of transcription, and the process of replication usually happen in the cell's DNA, cell's nucleus for the eukaryotic cells. So it says the information flows in one direction. So that's called the central dogma. It means from DNA to RNA and to the protein. So the central dogma includes three processes. The processes are one, replication, which I already explained to you before. And the second one is a transcription, which means writing down, converting the DNA to RNA. And the third one is the translation. So the DNA, which is already converted to the RNA, is being translated to be a protein. So RNA is said to be the link between the DNA and the proteins. There is something that is a bridge. The RNA is the bridge which connects the DNA to a protein, because the DNA cannot become a protein by itself. RNA differs from DNA in three major ways. One of it is RNA is called ribonucleic acid, not deoxyribonucleic acid. What does that mean? The meaning is the RNA has a ribose sugar, while the DNA has deoxyribose sugar. So the meaning of deoxyribose sugar is one oxygen molecule is being removed from the DNA compared to the RNA, which has a ribose sugar. And the second thing is, in RNA, we have thymine, adenine, guanine, and cytosine. So, but in the case of RNA, we're gonna have uracil replacing the thymine. So instead of you having T pairing with A, you're gonna have U pairing with pairing with A instead of T. So this is the second difference. And the third difference is RNA is a single strand, while the DNA is a double strand. So there is no, the RNA is a single strand, but the DNA is a double helix. So this is the structure of uracil. Now the transcription process is going to produce three types of RNA. The three types of RNA we have during the transcription process, which copies the DNA to make a strand of RNA, you're going to have mRNA, tRNA and rRNA. mRNA is called messenger RNA, tRNA is called transport RNA or transfer RNA, and the rRNA is called the ribosomal RNA. All of these three RNAs have a specific function they do. We're going to talk about that later. Transcription is catalyzed, means it's been made to happen at a faster rate by what we call the RNA polymerase. The RNA polymerase and some other proteins form a transcription complex, which is going to be the one that actually tells the the proteins where to start the unwinding process of the DNA. So the transcription complex recognizes the start of the gene and it unwinds it. So all of these things you see here is meant to be the complex. This is a transcription complex. So it is the transcription complex that is going to tell the enzyme, which is known as helicase, that helps to unwind the double helix. It tells the start point and it tells the end point. So what would happen is when the process is starting here, the free floating nucleotides, as you can see them here, they will be sticking, they will be finding the complementary bases, and they will be forming a double helix with it. So, but the only difference is instead of T pairing with A, you're going to have U pairing with A. So the transcription complex will recognize the start of the gene, and it's going to unwind it, and it starts the unwinding process. So nucleotides pair with one strand of DNA. The nucleotide is going to pair with one strand of the DNA, whereby the mayonnaise, you're going to have the free floating nucleotide pairing with one side. Now you've got two sides of the DNA. This is one part of the DNA. This is another part. So now you're going to have the free floating nucleotide. They're not ready to stick to both sides. They're going to stick to only one side, because what they want to do is they want to produce RNA from this DNA, and they want the DNA to remain itself after the production of the RNA. So what would happen is they're going to use one side, either using the lower side or the upper side. So what would happen is the free floating nucleotides will find a complementary basis maybe in the down section or the upper section, and it's going to form an RNA molecule with it. So the RNA polymerase bonds the nucleotide together, and the DNA helix once again, and the gene is transcribed. So this is the process whereby the free floating nucleotide pair up with the one with the lower section. So as that happens, now the process of the transcription is completed. As you can see, the RNA is being produced and is moving out of the DNA. So when the RNA moves out of it, then the DNA would wind up again and become the normal double helix. So this is what we saw here. So the RNA strand would detach off from the DNA once the gene is already transcribed. So once the gene transcription is done, you're going to have a DNA form back again. So this is the process of transcription. Transcription makes three types of RNA, as I mentioned to you before. The messenger RNA, it is the one that carries all the messages which need to be translated to form a protein. While the ribosomal RNA form part of the ribosomes where the proteins are being made. So you're going to talk about that in translation, whereby the two types of ribosomes we have. The transfer RNA is the one that brings amino acid from the cytoplasm to the ribosomes. Remember that in the lower grade biology, we talked about ribosome as the one that helps in protein synthesis. So we're going to talk about that in more detail in translation. The transcription process is similar to replication in some ways and is also different in some other ways. Transcription and replication both involve complex enzymes. Remember, the replication uses the DNA polymerase, but the transcription uses the RNA polymerase. So, and also the two processes are different end result. The replication copies the DNA, while the transcription copies only a gene. The DNA remains intact. A replication makes one copy, while the transcription makes many copies. So you can have only one copy when you replicate. You can have, like what I mean by this is, you have a DNA, you can have another DNA, you cannot have more than one copy at a time. So you can, well, except if you have multiple replication taking place. But in the case of transcription, you can have the process of transcription happening over and over and over again until you can make as many as possible copies of the RNA you want to make. But the replication is once you have a half of a DNA, you only have another half to make another DNA. So this is a picture that shows you this is one gene here, and this is a DNA, and these are growing RNA strands from the DNA. So this is exactly what transcription is all about. Thanks everyone for listening.